Here we are at 16.2, and we just spoke about the structure of DNA and its discovery. So now let's talk about how DNA is replicated. After Watson and Crick wrote their famous paper, which if you were in honors bio, you read this. They at the end of the paper they wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material." So even just the way it's made, it screams out like there's a copying mechanism. And so they proposed that the model was split in half. So if you have your two sides of DNA, that they're split in half, and one side is copied, and then the other side is copied, and then you have two separate strands of DNA, which we know as a semi-conservative approach. However, there were other scientists that had other ideas. There was a conservative model. So if you see here, you have one original strand of DNA, and the strand is conserved all the way through, that there is one original DNA strand, and it's copied, it's copied, it's copied. But there is no there's no pieces of the original strand included in any of the replicated versions. So we talked about the semi-conservative model, which is broken in half, and so this molecule has half the strand, the original strand, and this side has half the original strand. The dispersive model shows that the DNA is broken apart and distributed throughout, and that each molecule is a little bit of new and a little bit of old. And so Michelson and Stahl, here is a link. I will put this on PowerSchool for you guys to understand the Michelson-Stahl experiment, which could very well show up on an AP test. This is how they determined that it was a semi-conservative model. They did they put radioactive markers in the first replication, and then discovered what was present in the second replication. Once again, another very elegant experiment. So let's start off by knowing that DNA is cool. Our 46 chromosomes have 6 billion base pairs, and if the pieces, if the A's, the T's, the C's, and the G's were the size of the print in our textbook, it would be 1,400 biology books, which is so cool that we have that in every single one of our 5 to 50 trillion cells, 6 feet of it. However, that entire genome can be copied inside of the cell within just a couple of hours, with errors being 1 in 10 billion. So one error out of about 10 billion base pairs are mutations, which is pretty cool. So let's talk about DNA and where it begins. It begins at the origin of replication. The origin of replication, let's look at this bacterial plasmid, or a strand of DNA in the bacteria is a ring. It has a single ring-shaped a single ring-shaped DNA strand. And so here the origin is going to be like here and here, and then they're working out sideways. So you can see the little arrows showing that the enzymes are working and copying in this direction. They're splitting it apart and copying this as the replication bubble gets bigger and bigger. So this is the replication bubble. You can see that sort of here as well. So your origin of replication is here, and you're working out that way. And so we said that bacteria has a single ring of DNA called a plasmid. But in eukaryotic cells, we have 46 chromosomes, and on each chromosome, we're going to have hundreds to thousands of replication bubbles happening. This is going to, because we have so much genetic material, it's going to happen much quicker this way. And so let's look here, this Y-shaped region. This is the side of the replication bubble. So here's a replication bubble, and here our players are going to be helicase is splitting, is sort of increasing, pushing that replication bubble to get bigger and bigger and bigger. Let's see, like this, like this, like this, like this. It's splitting that bubble apart. So it's unzipping the DNA and those hydrogen bonds. And then the SSBPs, or the single-strand binding proteins, are proteins that attach to the nucleotides that are recently just opened so that they don't attract the other side and close up again. So for a moment, they're going to be covered so that they don't reattach. And then we have topoisomerase over here. So if you can picture like a string, and you know how it's kind of wound with other like fibrils, and if you pull those little pieces of the string apart, you're going to create more tension further down the string. And so what is happening here, topoisomerase is going to relieve the strain by breaking the DNA, swiveling it around, rejoining DNA strands to try and make sure that it doesn't get all like knotted up. It's relieving that strain of sort of pulling, because it's untwisting and pulling the DNA, and so it is going to relieve that strain. So enzymes, the enzymes that we're going to be talking about and that we have been talking about, cannot synthesize a brand new DNA molecule. They can only add on to an existing chain. So it's an interesting thought to think that the original molecule of life was, we believe it was an RNA molecule, and that we are still just sort of adding on to that original RNA molecule. And so DNA exists in all of us, and so there's still some of that original language in all of us. And so life comes from life. New DNA strands are not synthesized. Life is really interesting. So here we have, let's say that this is just our open strand, our open template strand. It's been split apart, and we're going to talk about primer or primase. So primase is an enzyme that's going to first lay down in a 5' to 3' direction, always adding on to the 3' end. Primer is added on, so it's an RNA, and it needs to be there so that other enzymes can sort of grab onto the molecule and start working. So without the primer, other molecules can't really attach. And so the DNA is split open, and then primer is laid down by primase. And so remember that it is going to lay down in a 5' to 3' direction. There are several different DNA polymerases in a eukaryotic cell. However, there are only 2 in bacteria. So let's study the bacteria. So DNA polymerase is the molecule that is going to attach onto that primer and then start attaching nucleotides onto the template strand. So we're going to have that semi-conserved. So here's our green, here's our original model, or our original strand, and here's our, so the conserved original strand, and here's our new growing strand. And so DNA polymerase 3 is adding on to that, that growing strand, always adding on to the 3' end. So in bacteria, the rate is about 500 base pairs per second. In humans, it's about 50. So we're a little bit more slow, but we are a little bit more efficient, or a little bit more effective, I'd say. So something new here for you guys is that a nucleotide, we've always studied it as a phosphate, a sugar, and a base. A phosphate, a sugar, and a base. However, I'm going to blow your minds here that a nucleotide is actually a triphosphate. Just like ATP is a nucleotide, it is a triphosphate. And so as we're adding on ATPs, or as we're adding on nucleotides, they are DATPs. So here is just a standard ATP that we know and love, talking about cell respiration and pretty much anything. And here is our DATP, or a nucleotide, our phosphate, our sugar, and our base. The only difference is that an ATP has a ribose sugar and DNA has a deoxyribose. And then our bases are going to be variable, A's, D's, T's, and C's. And so they are similar to ATP in that there are still 3 phosphates, and they are still 3 negatively charged molecules that are clumped together, and it's going to have a lot of energy when they are released because it is not stable. And so as DNA polymerase catalyzes the addition of the DATP, or the nucleotide, onto the growing strand of the DNA, it is going to cleave off 2 phosphates. And energy is released that helps to fuel DNA replication. We said that there's lots of energy. And that's going to cleave off a pyrophosphate. A pyrophosphate is a 2-phosphate molecule. And so what actually happens is that pyrophosphate is even further cleaved. It is hydrolyzed into single organic phosphates. And that hydrolysis is thermodynamically favorable and exergonic, and therefore is going to also help to fuel the process of DNA replication. And so we've been talking about replication. Let's talk about elongation here. So DNA polymerase is going to continue to add on to the leading strand. So here, if we look at this, we read in a 3' to 5', but we build in a 5' to 3', always adding on to the 3 end. Always adding on to the 3 end. So here's our leading strand. However, if we notice, since the sides are anti-parallel, this is a 3' to 5'. The other side of our conserved molecule is going to be 5' to 3'. And so if DNA polymerase 3 can only add on in a 3' to 5', can only read, in a 3' to 5' direction, it's going to have to add on in segments. And so we call those segments Okazaki fragments. This is called the lagging strand. It's going to be copied discontinuously, and the Okazaki fragments in bacteria are 1,000 to about 2,000 base pairs long. In eukaryotes, they're about 100 to 200 base pairs long. So let's just recap here, because there's a lot going on. So here's our origin of replication, and our leading strand is going to be, you can see here, a primer was laid down by primase, and here's our leading strand going in this direction. Here's our replication fork that's being split by helicase. Our SSBPs are here. So here's our leading strand, and here's our lagging strand. And so as this side is being laid here, and then here, you can see the primer on each of these Okazaki fragments. So we're just looking at this half of the molecule right now. Here's our leading strand. Here's our lagging strand being added on like this. And so if we look at the whole replication bubble, DNA polymerase is working in this direction, and there's a second DNA polymerase working in this direction. And so as it lays down this leading strand, it's going to be laying down Okazaki fragments on the other side. Let's look at what this actually looks like. So we often think of DNA replication happening with enzymes kind of driving down the DNA like cars on a road. But here's our DNA polymerase 3. It's actually a complex of many different enzymes. And so here we can see, when we start the video, we can see that here's our leading strand being synthesized continuously. But over here we can see our Okazaki fragments being made discontinuously, and then they meet up, and then it's starting over here again, and it's being synthesized that way. So here's our last slide. After the Okazaki fragments have been laid down, the primer needs to be replaced. So DNA polymerase will replace the RNA fragments with DNA fragments or DNA nucleotides. And so they're not actually bonded yet. What ligase does, DNA ligase, will then attach them, we'll bond them, and create that sugar phosphate backbone. All right, so that's 16.2.